Rindy. Thanks. Thanks everyone for joining us today on this webinar. My name is Gustavo Teixeira. I'm leading the Breeding Operation and Phenotyping module at Excellence in Breeding. Our goal in our target in Excellence in Breeding is that all the public breeding programs for CGIAR, the national programs, the partners that we have a high rate of genetic gain and really modernize to achieve that. In order to do that, we have some modules and from product profiles, definition and market segment, breeding schemes, module 2, module 3, that is genotyping, this is in EIB. And the module 4, that module that I lead, we target the improvements in operations and phenotyping. This targeting the good data, how to get a better data, how to have a sustainable operation, etc. In order to do that, or how to improve that, we have some initiatives, some strategy. But one thing that we realized is the need to share practice. We don't need to. First, we don't have capacity to support everything. So to do everything, so we need to interact. We need to create this, to collaborate. In early or mid this year, we launched one program called BOND, that is the Breeding Operation Network for Development. The idea for this network is to connect the operational people, the people from different national programs, from different CGIAR centers, from different companies, to collaborate and to share good practice, to share knowledge, and in our idea is that with that these things the improvement will come, because things need to happen at the operational level. We have some pillars on this bond. The first pillar is the Bond 101. So what we call Bond 101 is where we have from our team in EIB, and of course with support from some consultants, some partners, we work close with some operational staff from different institutions to support them to improve, to implement some projects. This Bond 101 we started in June this year, July. We have now 65 to 70 members in this Bond 101 from different institutions, from CIP, from CIET, from IITA, from IRI, from CIMIT, from NARO, from CSIR, from SARI, from Africa Rice. So we have members from different institutions, from ICARDA, and these 6-7 members, they are organized in 2 groups. One group that are working with improvements on different projects related to material handling, seed process, material handling. And another group is now working on projects related to irrigations, to drought screening trials, to how to manage irrigation. So with these groups, we have total now something around 10 to 13 projects that we are helping them to implement, and projects that are basic projects, things like, for instance, how to improve how to organize our seed storage area or whatever. So different type of projects. So this is the Bond 101. The other pillar for our Bond program is the Bond webinars. So this year we organized 5 webinars from different topics, from health and safety, from seed multiplication, from phenotyping, etc. And in today, I understand that sometimes, even though that the webinars that we organize and independent of crops is much more thinking on behavior, thinking on practices, but of course some techniques are specific for different crops. And today this webinar with Ardevo is a good opportunity to, especially for RTB crops. Once this can also and Julian and the team will present today. I hope that this will help some breeding programs, especially, of course, this applies for everyone, but especially for the potato, for yams, for cassava breeding programs, to see different operational ways of work. So this is what we hope, and today is the last one for this year. Next year we already have some plan to bring other webinars. Yeah, so this is the second pillar. We have other two pillars that we are still working on. We hope that we will put some materials, some e-learning materials in our platform that people can access for training, and also to organize some technical visits. Of course, these technical visits, we depend on the situation that is uncertain now. Yeah, with that, we hope by doing that, we hope this will give more accountability and more develop the capacity internal for these breeding programs to increase the use of mechanization, to increase the automation, improving the practices, and by doing that, have a more a better process with that the process is most effective, cost efficient, would work in a safe workplace, and of course with the goal to have a quality data. Okay, so with that, I'd like to thank you for joining us today. As I said, I will have two speakers from the webinar. Julian is a potato breeder from Ardevo. Prior to join Ardevo, she was also a CGIAR breeder working with SIP. And we also have Tim, that is the trial manager for Ardevo, with lots of experience in potatoes and managing trials, managing different potato operations for potato, not only breeding, but also in some production. With that, thanks, Julian. The floor is yours. Thank you.
Thank you. I will share my screen. Okay. So first I want to say thank you for the invitation. I was very happy to tell you more about what we're doing at Ardevo, and especially because of the work I've been doing with SIP before. This is a completely different environment, but still we are facing similar issues, and we are handling an RTB crop as well. So I'm very happy to share this information with you today. So let's start. These are the topics I will touch today. So I will start with an introduction of Ardevo, then go to good practices in potato seed production. This will be presented by my colleague, Tim. And then I'll continue in the breeding areas. So first, a little bit of context, how traditional potato breeding is done. then going into good practices from the field preparation to data collection, quality control measures, data management and seed inventory. And then I will end with an overview of how Ardevo is moving towards a hybrid seed crop. So first, Ardevo, we are based here in the Netherlands, and it's quite a young company. So it started as a research project from KWS. If hybrid potato, if it's possible, what are the possibilities, and is it, yeah, how can we move into that direction? And that started in 2012, and then after a few years, they have developed a joint venture, and that's called Ardevo, between KWS and Simplot, and it started in 2018. So we are a young company, but we have the advantage of this shared experience between the hybrid breeding in KWS, but also the processing industry from Simplot, and that's a combined experience of 250 years. So we are based in the Netherlands, here in Nagelen. We have now a team of 20 people doing the main thinking work, let's say. And then the technicians, we are based on KWS sites, we can use their support. And the same is true for the US, where we are based at the Simplot campus in Boise. We have a smaller team there, 3 breeders, also working on hybrid potato. So for Ardevo, we are focused on 2 segments. In Europe, we have the French fries and the crisp, and in North America, the French fries. And this was really to help us moving into a direction. That means that for these 2 segments, the demands from the industry are very clear, and then we can make sure we are targeting those standards. When we come up with a hybrid variety, we can compete in those markets. So when I talk about potato, I think for this kind of audience, we have to be clear that to be able to produce potato, we have to go through a lot of multiplication steps. So it is a clonal propagated crop. It means that we have to produce seed potato first, and using that seed potato, that's the input for the ware potato production. And it takes a lot of logistics and also a lot of handling. Well, the whole sector around it is very specific to the seed potato production. And that's where my colleague will tell you more about.
Yeah, thank you, Jolien, for the introduction. And yeah, I would like to talk a little bit more about this, let's say, clonal propagation. Yeah, so this is normally done in seed potato production. Yeah, I have included a picture where they are being exported to. So they are going all across the world, going to many different countries, many different markets. But they all have in common that it's really important to have high quality seed potatoes. In fact, it is impossible to grow healthy potatoes without these high quality seed potatoes. Yeah, so the seed potatoes are exported to all parts of the world. And you can see from the image that a lot of them have their origin in the Netherlands. And I would like to talk a little bit more about how the operations are being done in the Netherlands. So yeah, my part of this presentation will be mostly, I would say, only about seed potato production. And I will talk less about the ware potato production. So it's yeah, really important to make this distinction. I will be talking about seed potatoes. For the seed potatoes, there are rules in the European Union that are equal to all the member states. They have to comply. But in addition, the countries can place more stricter requirements on the material. And take Netherlands for example, and there are other countries might have different things, but in the Netherlands, there is a maximum potato share in our crop rotation scheme. And yeah, this is checked, all the requirements that the seed potatoes have to fulfill. They are checked by the Minister of Agriculture and Nature and Food Quality. That is a government organization, but the inspections are done by an independent organization. So that is not a government agency, and that is called the NAC. Yeah, it's translated to the Dutch Inspection Service for Agricultural Seeds and Seed Potatoes. In the Netherlands, the seed potato cultivation is concentrated close by the sea. And there's a reason for that, because the main things in the potato seed production is to keep the crop healthy. And the threat for that is mainly virus and bacteria. Virus is mainly transmitted by aphids. So if you have a low aphid pressure, you have a less risk for virus in your material. And that is why the fields are concentrated close by the sea. There are less aphids close by the sea. The seed potato growers, they are mostly members of a trade company. These are very large companies, and that can be either a corporation, or it can be that they are more independent, more commercial. But in addition to the large ones, there are also a number of smaller ones, and they are owners of specific varieties, and also they are growing varieties that are not that do not have an owner, let's say. The seed potato growers are highly qualified specialists. It takes a lot of skill to grow the seed potatoes with high quality, and there are many varieties, and farmers are quite often specialized in a number of varieties. In the production, the number of seed potato is more important than the total yield. It's really, from one tuber, it's one plant. So it's more about the number than about the size. So this is the opposite of rare potatoes, consumption potatoes. Then there is a short season because it is about the number. So the seed potatoes, they are in principle, they are a smaller size. The size is between 28 millimeters and 55 millimeters. So that means the season is also shorter to keep the potatoes small. And this has an added benefit that you can escape the aphid pressure, which builds up more later in the season. This is an overview of the system that is used in the Netherlands and also other countries in the European Union. It works this way, that it starts with tissue culture, PBTC, and that is the generation 0. They are being propagated every year. And by doing that, there is a multiplication factor of 10. So that is in terms of volume, but also in the size of the area, the number of hectares. So every year, you go down this ladder, let's say, and you go to the next generation. And there is also a quality class attached to that. So normally it corresponds to the generation, but it can also be downgraded or even rejected when the quality is not there. Can also see that there are different colors for pre-basic, basic, and certified. This also corresponds to, let's say, different farmers. They specialize in a specific type of seed potato growing. So the pre-basic is, these are specialized farmers, and also the basic seeds are specialized farmers. And the A and B quality, that is the potatoes that you can buy as certified seeds that you can use. The reason to do the multiplication is mainly driven by cost. Yeah, every time you multiply it, the cost of the seed potatoes go down. Yeah, then during the season, yeah, to maintain this quality, yeah, there are 3 different things that are being done by the independent inspection service that I talked about previously, the NAC. Yeah, they do 3 things. First, they start with field inspection, and they will visit the farms throughout the season. And they do the inspection visually. So they go into the field and they look at the plants, if they're healthy or not. And in this way, they can see the entire field. So they can have a lot of information about the health status of the potatoes. Yeah, so this goes by eye visually. Yeah, you can see a lot of potatoes, but there are also, at the same time, there are limits for that. That virus has an incubation time. So especially, yeah, at the end of the season might be already stopped, but the plants are already infected. And you still end up with the virus inside your tubers, but you cannot see it anymore. And you cannot see it in the field because of this incubation time. And for that reason, there is also a post-harvest test. And that is a laboratory test, where they take a sample from the storage, and then they can they check the percentage of the virus. And they also check the bacterial disease if it is a quarantine disease. In addition to that, there's the third thing that they do is the lot inspection. So that is really in the end, when the potatoes are being made ready for export, you have a final yeah, look at what is in storage and what are they transporting. Yeah, and then they look for also, it's again a visual inspection. So inspection season starts in spring with the registration of plots and starting material used. So all the plots, they are being declared where they are exactly. So it's an online system that the farmer needs to apply their fields to. So the inspectors know exactly which fields they need to inspect. And you also get the certificate in the end on that field. So you cannot use another field for that. Yeah, the seed potatoes are inspected during the entire growing season. Special attention to tuber-borne disease, general appearance and development, and varietal purity. Then we get to the end of the season, and then it's necessary to destroy the foliage to prevent a possible virus infection into reaching the tubers. And for this, they kill the potatoes by a combined action of mechanical and chemical destruction. This also has an advantage of keeping the tubers small if you do it before they're fully developed. Yeah, you have the quality and also the right size. And after that, there's the post-harvest test for virus and bacteria. Then we have the lot inspection. They look for quarantine disease, cereal disease, external defects, shape and sprouting. So there's the total weight of the total lot. And this has to correspond to the field, so you cannot add extra potatoes. Yeah, and look at the tuber size, specific requirements for that, and contamination. Yeah, mainly related to soil, all types of contamination. And the farms are being visited daily during the grading. So they have a complete overview of what is in storage. Now I'll talk a little bit more about what we are doing in as Aardevo. So for us, it's also important to maintain a good quality of our fields. It's because we have the genetics and the environmental interaction, and up top of that, there's the field management. So we would like to have a good quality for all our trials. And also we want this management to be similar across all our trials, so we can make a good comparison between the fields, and we can test in that way the genetics. And also we are interested in the environment that they are in, if they are more specific for us, the kind of environment, or that they are for more yield stability, that they have good yields across multiple locations. Our target growing areas are, it's now for this moment, it's France, the UK, and more Western Europe and the Netherlands. We test different soil types and subclimates, and we are doing yield trials. We look at how much yield we get with a good quality. We start in the Netherlands because we use a new system, so it's not really grown from seed potatoes, but we use from seedlings. So we first grow them inside a greenhouse located in the Netherlands called Gidsels, and there we let them germinate and let them grow to a small plant. So they are later on, they are also able to survive the field conditions. So first they have several weeks of growing inside the greenhouse before they are being transported and transplanted into the fields. So they start at the same location, but they end up at different locations. So you can see some of our trial fields, yield trials. They are from the Netherlands and in France. You can see also different soil types that we test, different climate conditions. We test all the varieties that we make. We test it out in the field.
Great, thanks, Tim. So I will continue with more of the breeding point of view. So before I go into details on the logistics, I think it's good to give some context on how potato breeding is traditionally done. So a new variety is created by taking two parents, crossing, and evaluating the offspring. So it means that every cross produces a lot of seedlings, and that's usually done in just one location and small plots. And every time or every year, a selection is done. The best ones go through to the next season in larger plots, going to more locations. And what comes out after many, many years, so that's here at year 8, that's the potential variety that goes to registration. But if you look at this process, it means that the variety where you end is coming from one single plant in the beginning. So it is very important to trace that process, to know exactly this is the pedigree, this is the genetics that I'm working with, and my genotype here is going through the selection steps and becoming a variety. So by going through all those steps, we also need to multiply our material, keep it healthy, and being possible to evaluate in different locations, just what Tim explained. So everything that happens in the industry, we also have to follow the same regulations, the same rules to be able to evaluate the material. And if we go from one step to another, we have to work with nature. So there are limitations to every step. And in potato, some things you cannot change. You have to break dormancy, and that when the tubers are harvested in the field, before we use them in our program to cross, we know that we have to break dormancy, and that takes on average 6 weeks. Then when you see flowers, after pollination, it takes time to set the berries. So here, again, 6 weeks before we can harvest the berries in the greenhouse. Then once we have the seed, we can sow them, and then it takes another 6 weeks to raise the seedlings. So for us in our breeding scheme, it's quite important to have a strict deadline or to have a strict planning scheme where we take into account these limitations or restrictions. That's because you cannot force the plant. You can force them with some ways, but we need to respect the timelines. So in that processing flow, keeping track of the material is very important. We can do that by digital data collection. And here at Ardevo, we use Philbook. I think that's quite known among this group. So we are using barcodes, and every plot, every single tuber has to have a label, knowing exactly where it comes from, what genotype it is, and which stage it is, and where it goes to which program. So capturing that data in Philbook is a way of avoiding mistakes. It's standardizing the data collection and using the trait ontology that helps to standardize the way of capturing data, and the traits you can compare among those locations and among years as well. So when the material comes from the field, we harvest, phenotyping is key, and that is true for any breeding program. We need to phenotype accurately, and we can do that in a destructive way or a non-destructive way. And also we can do that high throughput or low throughput. And that's some progress we're trying to make to have more non-destructive measures. So then it means you can still use the tuber for other purposes. And of course, we want to have high throughput, having data as fast as possible to make decisions as fast as possible. So given an example, looking at the tubers, we can use a camera to look at the length-width ratio, or we can do that, of course, manually, and then it's low throughput. So finding which traits we can do in which way is very important of having that accurate data on time. And the quality of the data also depends on the practices in the field. When we bring the tubers to the field, we can make a field layout, really optimizing the design with a row column design. We use the checks that are available. These are the varieties in the market. We use them as a benchmark. Of course, the health status of the planting material is very important, that all what you have in one field should be having a comparable health status. That means that also the checks in that program need to have a comparable health status. So the way you design the fields can help in quality control. Just an example here is that every plot can be separated by a different genotype, which gives a red colored potato. And as you see in the picture, visually in the field, you can already separate one genotype from the next one. And also the planting distance can be very specific to the needs of a program. It means that if you have to evaluate lots of seedlings, they can be planted at a different planting distance. Then you go to much more advanced material where we go to look at different traits. And also for us in Ardevo, we are looking into machine development specific for the harvest type. What I mean is that harvesting plots is depending on what do you want to do with it. Is this now material for multiplication? Is this now material that goes into processing? So we have specific machinery that's helping in that way. So when we talk about quality control, what you can see visually, the phenotype is only one step. So in the field at harvest, you can look at a plot, collect the tubers, and these tubers should look the same because it's one genotype, for example. And then bringing them to the barn to storage. After that, these different tubers can go in different directions. So one tuber can go for multiplication, another for crossing, another for a different location. And by keeping track of that with barcodes, the next season, we expect that these genotypes are still the same. But that's not always the case. And I think a quality measure in between that is very key to avoid a mixing. So that means that you think it's the same genotype, but in practice, it's something else. Something can happen along the way. Every handling that happens, something can go differently. So there is a check with markers to say, okay, we want to check the identity of this and that. Is it really the same? Is that the genotype that we think it is? So we use an identity check on the genotypes used in our crossings. We also do a verification of the cross. So in the large crossing scheme, are we doing exactly as what the breeder wants? And then if that's not the case, we can check in our database, maybe from the plants nearby, this can be the parent. So we have a tool that can suggest what the parent can be if it's not what you think it is. For us, the diploid check is also important because we work only on diploid potato. So anything that's not diploid is thrown out. So also in our trials, we need to have control on health. So the virus status, we are working towards zero tolerance. And that means that any plant that is infected is removed. But I've also seen here that a visual assessment is not always correct. So there is a risk of throwing out material that was actually still good. And in potato, because we are working with different generations, it is important to, even in the field, to organize the different trials, not mixing the different generations. That means that a young seedling should be planted in separately from a tuber multiplication. And then walking through the field can be minimized, so not to transmit any disease. So data management and all of this process is very important. We have our trial design that's done with our program. And then the unique identifier follows the material along the way, because one genotype can be in different places or can be in different health status. So we really need to know exactly what is where and in what state. So the phenotyping data, like I explained, we are collecting with Philbook. The genotyping data is all collected in one central database. So we have all of our trials connected. And I think that's quite nice to have, is the seed inventory is linked to that. So it means that any tuber that goes in or out is registered. So if we need pollen, we need through potato seed, everything is in our database. We can exchange materials between programs, and we also know what is available, what is not. And then, of course, also the crossing schemes is all part of the central database. So a database can be more than just storing data. So for us, the data that's in there is the program, the number of entries, the season, the location. But we also use it as a planning tool. And so there is a way of saying, okay, this program is ready for planting or can be harvested. This is where the tubers go. So the planning of the season is linked to the database. It's also a budgeting tool. So we know how many plants there are. We can calculate next year, this is what we need. And then it's supporting us in making those plans. And the communication tool, because we are working together with KWS with their technicians. They are using the same system. So exactly what is going on is all there in the database. So then I will come to the more specifics of moving towards a seed crop through potato seed and potato. Yeah, that's potato. Is something that SIP has been doing for a long time already. There has been a lot of research. So there is a lot of knowledge there, but it has not changed the industry up to now. And there are reasons for that. So through potato seed, you get from a biparental cross. You think these are good parents, you cross, you get seed. And the seed is especially of importance in environments where the access to seed potato is very limited. So there, through potato seed has played an important role. But for now, the yields are lower when you grow directly from TPS instead of seed potato. So to generalize a bit, TPS can be successful, but there are 3 conditions you need to have. So the, TPS can make it possible to produce hybrids and exploiting heterosis. You need to have a homozygous product, and that's you can do by crossing inbred lines, and also the seedling figure needs to be good enough. So at Ardevo, we are transforming from a traditional breeding to hybrid breeding, and that's not going overnight. We need to go through different steps. So from tetraploid, we are changing to diploid potato. Then once we have a good diploid material, we are producing inbred lines, and by combining inbred lines, we can make hybrids. So this picture is a very simplified visualization to say traditional breeding, it's very complex. You have 4 chromosomes each, you have a lot of segregation, and the offspring is very, very much diverse. In hybrid breeding, you go to inbred lines of diploid material. By crossing those, you get a uniform hybrids or uniform plants. So when we talk about hybrid potato, we talk about 2 innovations and 2 models. So first, we are producing hybrid seed, and that hybrid seed can be used to produce seed potatoes. And seed potatoes can grow and produce the wear crop. So the last step from a seed potato to commercial wear crop is not different from what's happening today. So that's the same system. Only thing changing is that we can go from seed to seed potato much quicker. In the long term, we want to go straight from the seed through potato seed to a wear crop, but that will take a bit more time because it has other innovations that need to follow. And these are more agronomic innovations like optimizing the growing conditions, optimizing the way we transplant, and so on. So 2 innovations, there is a genetic innovation and also an agronomic innovation. So why do we want to go to TPS? There are some advantages which are very clear. We can produce we have a much higher multiplication step. Seeds you can preserve longer, and there is a limited transmission of diseases via TPS. And also the logistics is much more easier to do. You replace a full container of seed potatoes with a bag or an envelope of seed. So there are a lot of advantages, and that's where we want to go. So the question is, where are we now? So do we have the seed? How close are we to that new model? So here's a picture of me in the field in Naarden in the Netherlands with our first hybrids in the field. You can be very skeptic and see that the picture shows you that the hybrid is not uniform at this moment, but we were already very happy with the, just to prove that the concept of hybrid breeding works. We can produce new tubers from a cross between inbred lines. And the pictures on the right is to show you our diploid material, or starting material, that's now up to the level of the tetraploids. The tetraploids where we are, that is our benchmark finally. We are not there yet. There are no hybrid potatoes in the markets, or we as Ardevo, we don't have them. We are working towards it. So we still need a couple of years, and we are continuously improving. And that's because we need to improve the breeding strategy. It's a completely different system. It's making hybrids. It's also making different pools, making inbreds, and there is much more to optimize along the way. We are always improving our phenotyping methods, because the more accurate you get, the more you learn about it, and the more focus we can, or the more progress we can make. And then we go into the area of the seed production systems, where we need to produce actual seed, and growing inbreds is part of it, optimizing that production of seed and the crossing methods. So for us, the resistances is something, a must-have, let's say, in new varieties. So in diploid material, we are able to do a lot more backcrossing to introduce these resistances in our lines. And then as last, the TPS agronomy is it's a new topic. We need to find out what are the agronomic challenges, how can we do the pollination, how can we do the seed production, and growing seedlings is a completely new area for potato. But there are examples in tomato or other crops, let's say, where we are learning from, and so we can optimize our own system. So I want to end here. Thanks a lot. I hope I gave you some new insights in potato breeding and potato logistics. Any questions I would like to answer.
Thanks, Julian. Thanks, team. Really, I'm pretty sure that it helped a lot. We have some questions in the chat. The first question is from Anil Kumar. He asked how many clone you took every year from 3 years of process. Is slide 17.
Yeah, so you start with many seedlings, that goes into 10,000s, and then you reduce. So every breeding program has its own optimization, I would say. So what happens is that the number of lines reduces, and that means you can go to more locations, more plots. So exactly how many, it depends on the size of the program and the stage where we are. So it's hard to give a definite number there.
Mm-hmm. Thanks. Another question also from Anil Kumar. Is it necessary to use border plants to separate plots? Why don't leave some space between plots?
Sure, there is space between plots, but we have seen that that is not enough, because the plants tend to grow under the ground, and especially when we use machinery, it tends to. It's a quite difficult issue for in general to keep plots separated, and these visual separators are really helping us. So I think it's a nice idea to it adds space, but it also has a lot of advantages.
Thank you. Another question here from Teresa. Did your company create the breeding software, or is the program software available commercially?
We are lucky, I would say, that we can work with KWS, so we use their system at the moment. So that was already useful in other crops, and it was ready for other crops, and we have made some adjustments to make it work on potato. So,
yeah, a question from Bodo. Where do you see yourself relative to Solinta?
Yeah, Solinta is a spin-off, I would say, from Wageningen University here in the Netherlands, and they are also completely focused on hybrid diploid breeding. Solinta is, correct me if I'm wrong, right? Because I don't work for Solinta, but they are funded by working in projects and more targeted towards other segments, other areas. So we have our own
market
strategy. We work towards the European market, so that differentiates us from Solinta a bit more.
Yeah, Mokhtar asked, how do you plan to define the parents selected from the inbreeding scheme?
Yeah, so hybrid breeding, we have developed different pools that are heterotic. So we mean that by combining them, we expect some heterosis. So we have trials looking on GCA, SCA, and then that's how we build up our pools, and then how we improve the material every cycle, trying to get better material, and that's the input for the inbreeding part. That's
thank you. Yeah, thank you. I see one more question here from Same. Can we start the breeding program with tetraploid parents?
Well, there are many advantages of going to diploid potato, and one of them is that we can go to homozygous lines. In tetraploid material, this is it's not feasible, because every time you have inbreeding depression, you get weaker plants, and if you have to go to many generations before going to a homozygous plant, it's just not feasible. But there are also other advantages of working diploid potato. We can bring in other resistances, and there's much more knowledge, and much easier to work with on the marker level and so on. So it is possible to work with hybrids and tetraploids, but you won't be as effective.
Yeah, thanks, Julian. We have another question here from Sira. Many thanks for the excellent presentation. How different is your hybrid scheme relative to that used in maize breeding? What lessons should RTB breeders learn from your inbred strategy?
Well, to answer the first question, it is completely the same. We are doing the same as in maize. In maize, it has also taken a periods where the inbreds were difficult to grow and to optimize that production scheme. So we are following the same trends as in maize. So I think potato is just a little bit harder. They don't like inbreeding, so we have to find the right way to do it. So it is not as easy, but in practice, it is very comparable. So, and then the lessons you can learn from the inbred strategy, I would rather see it as a lesson from what clonal multiplication can do. I think we are combining two worlds here. We can go, we have the advantage of bringing the same genotype to different locations, plus we can produce the seed to go to a larger scale at a faster speed. So the RTB breeders, I think my lesson would be to you that keep an eye on the quality, keep an eye on the health of the plants, and because the agronomy is as much as important as the breeding progress.
Thanks, Julian, team. One question I have is, you moved from CGIAR, from breeding program CGIAR to a program in the private sector, and
I believe that is completely different environment for you two. Related to operations, related to what would be the advice that you can give to the CGIAR, to the NARES breeders, that you think you would be doing differently if you had learned that before? What can you maybe give us some advice?
I think the main difference from what I've seen here is really on planning. There is a strict plan. This is where we want to go. This is the material we get. You know, a plant can produce 8 to 10 tubers. What do I want to do with these 8 to 10 tubers? And to make maximum use of that and to plan ahead. Let's say these are the locations, this is where we need to go. I think that planning is much more structured from my experience, I would say. So that's something I learned coming here.
Yeah. Thanks. Yeah, definitely is something that we really need to improve, and many reasons why, but certainly we need to improve that, definitely. So we have time for one more question here. That's okay for you, Julian?
Of course.
Yeah. I see one question. What's your opinion on the recent patent applications on potato hybrids technology? Do you feel threatened by it in your work?
It's a good question. Yeah, we are aware of the patents that are along this hybrid breeding for potato, and not to give away all of the details, but we also know that it will take much more time for us to come up with something new. So the patent is there, and we have our own material, and we know what we can do and when we can do it. And I think I don't feel threatened. It's just so much more we need to develop and we need to work out. So it's a challenge for all of us. If there is a patent or not, we need to move forward on it. I think that's the future we need to go anyway.
Yeah, thanks, Julian. Yeah, we are running out of time here. I will ask you if probably there's more questions in the chat. I will review that later, and also, of course, also ask people to send questions to me or to EIB team. And Julian, we will collect that questions and send that to you. And if you have time, please respond that to us and send that, and we will publish that in our EIB website, and people can go there and read. And of course, if we have any specific questions that you prefer to respond directly to the participant, of course, it's also possible. Yeah, thanks again, Julian. Thanks, team. Thanks a lot for your time. Thanks for. I'm pretty sure it helped achieve what at least my expectation, and I'm pretty sure the expectation from the audience too. Thanks a lot. Have a really good day. I don't know if you have any final comments or Julian and team.
No, I think if people are interested, they can reach out. We are open for feedback or for anything that comes out of it. So I'm really happy to share, and it's something we all have to work on. So I'm
yeah.
Thanks,
Julian. Thanks a lot. Thanks, team. Pleasure. Yeah. Bye.
Bye.